Good afternoon participants, welcome to e-course on plant tissue culture. Today we are going to discuss on in vitro organogenesis in plants. Now let us first see the content, what we are going to see in today's session. What is in vitro organogenesis? Okay, that we will discuss. Then later on we will discuss what are the types of organogenesis, what are the different types of regeneration mechanisms which are available in any explant of a plant material. And what are the factors which affect shoot organogenesis and root organogenesis? We have already discussed in our previous session that there are three main factors. One is nutrient media, the other is plant growth regulator or the plant material, as well as the culture environment. So these factors, there are many other factors which also influence shoot organogenesis and root organogenesis. So we'll see those factors in this session. Again, we will see about somatic embryogenesis, and as I already told you that this somatic embryogenesis process has such a huge potential in making mass multiplication of any plant species, and that has already proven in many forestry systems as well as in many other medicinal plants as well. So we will see the factors which affect somatic embryogenesis as well. So what is organogenesis? The formation of root, shoot or floral buds from the cells under in vitro cultures in a manner similar to adventitious root or shoot formation in cuttings, in vegetatively propagated cuttings. It is also called as in vitro organogenesis. So in vegetative propagation, what we do is we take some shoots or we take some cuttings from medicinal plant, from horticulture plant, and we sow them or we bury them partly into the soil in a mini bags or in nursery bags. And then you get shoot formation from the axillary buds, and from the buried portion you will get root formation. So somewhat similar manner when you inoculate any explant, whether it is a leaf pieces, axillary buds, apical buds, root pieces, internodal stem, or any other plant material, the explant which when we inoculate on the media, and that media along with the cultural environmental conditions gives rise to shoot formation or root formation or any other organ formation like tubers or flowering. That is called as a organogenesis. Now plant displays extraordinary ability to regenerate complete shoot system from a tissue fragment or even from a cell. And in my previous section when we discuss about thin cell layer system, we have already seen that by using even epidermal cell layers, the cell layer has a capacity to form meristematic clusters, and these meristematic clusters has become predetermined to either form a shoot or root primordia, and these shoot and root primordia they can form either shoot or root. So plant system do have an extraordinary ability in that sense. Okay, organogenesis takes place in two stages, namely caulogenesis or shoot initiation, or rhizogenesis or root initiation. So these are the parallel terminology which are used in many books or even in many literature. So that's about in vitro organogenesis. Now shoot or root organogenesis, they are mainly controlled by plant growth regulators, which we also called hormones, which are we added in the media. And it was found that generally a high auxin to cytokine ratio induce here I should write root formation, but it is a high auxin will induce root formation, low cytokinin will induce shoot formation, and a high cytokinin will induce shoot formation, and low auxin will induce shoot formation. So it should be high cytokinin shoot and high cytokinin shoot and high auxin root formation. So organogenesis starts with the development of group of meristematic cells which are called meristemoids, which initiates primordia, and these primordia later on gives rise to a shoot formation. Whereas indirect organogenesis involves two phases of incubation. First is a callus induction on auxin rich medium, followed by shoot organogenesis on cytokinin rich medium. Okay, so indirect organogenesis has one additional phase, that it has to go through callus formation, and callus will later on depend upon the combination of plant growth regulator will able to form a shoot or root. Organogenesis or regeneration in vitro can be again divided into three types: indirect organogenesis, formation of organs, or direct formation of organs directly through callus formation. It is called indirect organogenesis. Direct organogenesis is basically production of direct buds or shoot buds from a tissue without no intervening callus stage. So there won't be any callus stage, callus induction stage in direct organogenesis. That is called in that stage it is called direct organogenesis. So whenever there is a no intervening callus formation, it is direct organogenesis. When there is a callus formation, it is called the explant which gives rise to callus, and callus gives rise to shoot, that is called as indirect organogenesis. And then there is a third type which is called somatic embryogenesis. And somatic embryogenesis is again it is able to induce from somatic or vegetative tissue a complete embryogenic callus which leads to embryo formation, and this embryo germinates to form complete plantlet. Now this somatic embryogenesis could be also direct somatic embryogenesis or indirect somatic. So in direct somatic embryogenesis you will have a lot of clusters of embryoids which are developed on your leaf surface or on the stem surface, and these embryos further proliferate and develops all stages which we have discussed in all last stages, that is globular, heart shape, torpedo shape, and then cotyledonary stage. And after cotyledonary stage, that particular embryo germinate to form a young seedling. That young seedling will have cotyledons, will have a hypocotyle, will have a epicotyle, and if it further proliferate, it will form a first leaf, second leaf, and so on. It will form a mature plantlet. So that is a direct somatic embryogenesis. In indirect somatic embryogenesis, the explant will first give rise to a nodular callus. A nodular callus will have an embryogenic potential, and therefore the nodular callus on a further media will form all types of embryoids, and these embryoids will again give rise to all those stages which I just said, and they will give rise to a complete plantlet. Now we'll discuss about direct organogenesis. Now each explant develops a meristemoid clusters which gives rise to either shoot or root primordia, which develops further to form a shoot or root. So explant can also form a root as well. So for example, if you put a leaf disc and you inoculate on auxin media, there are chances that that explant will directly give rise to even roots as well. Okay, so not necessarily that every explant will give rise to shoot first and then root. There is a chances that it depends upon the media combination and plant growth regulators that that particular explant will give rise to a root formation or shoot formation. It can give rise to root formation as well as shoot formation. So these obviously there is enhancement. So what happens? There is enhancement of axillary bud proliferation or elongation. Okay, in case of shoot formation, these axillary bud proliferation will also, there is a chances of adventitious shoot formation as well. There is a chances of adventitious root formation as well, and this all depends upon the auxin concentration and cytokinin concentration. If the auxin concentration is high, you will have likely chances that that particular explant or that particular shoot will form a in vitro rooting. Okay. Now, if the cytokinin concentration is really high, the pre-existing meristem which is present in axillary bud and in apical bud, it will give rise to shoot proliferation, or which we call as a shoot sprouting and elongation. That sprouting and elongation will generally happen on a higher cytokinin media. Okay, that will happen on a high cytokinin media. Now, along with this axillary meristem, as I already told you, that shoot from where the axillary pre-existing meristem is proliferating, that explant as well as internodal explant or leaf explant has a capacity to form a de novo meristem formation, newly formed meristem, and that is those are called adventitious meristem formation. These adventitious meristem formation will form new shoots. Okay, now when you have intermediate concentration of cytokinin and when you have intermediate concentration of auxin, there is a chances of callus formation. Now again, this callus depend upon the auxin and cytokinin concentration. If the cytokinin concentration is relatively high, then it will form a shoot formation. If it is the oxygen concentration is relatively high, it will the callus will give rise to in vitro rooting formation. So these are the different types of organogenetic phases will achieve depend upon the relative concentration of oxygens and cytokinins. And you can see this is a very nice photograph where you can see that if the BAP concentration, if it is 0, the explant is not showing any result. But as you increase the BAP concentration, which is benzyl adenine, it is a synthetic cytokinin. When you increase it, you will find a lots of shoots formation, and the frequency of shoots is also increasing higher and higher at increasing the concentration. Similarly, as you increase the indole acetic acid or naphthalene acetic acid, both IAA is a natural oxygen, whereas naphthalene acetic acid is a synthetic oxygen. And if you see the concentration, as we increase the concentration, there is an increased root formation as well as callus formation. Whereas if you increase the naphthalene acetic acid, it is also increasing the root formation. But if you see that if you increase the oxygen concentration and similarly you increase the cytokinin concentration, in that cases there is some mixture of shoot formation as well as callus formation. But this is a direct organogenesis. So you can see a variety of morphogenetic results in this particular photograph. So direct organogenesis, as I have shown this figure in my previous section, that if you take an explant, if you take this entire leaf and you cut a leaf pieces by using a surgical blade and blade holder, if you cut into rectangle, circular or whatever the shape, when you inoculate that in a media, on a media in cultural condition, first week it will swell, in second week it will start forming meristemoids at the cut portion or near the cut portion, and this meristemoid will give rise to smaller shoots which are adventitious in nature, and these smaller shoots will proliferate further and will form elongated shoots. So this is a direct organogenesis. This is again a diagrammatic representation of organogenesis, where you can see in this particular petri plate that there are lots of meristemoid formation and small shoots which are arising from the cut portion of the leaf. If you inoculate further or passage it further, subculture it further, you will get more adventitious shoots, and these adventitious shoots elongate further to form a complete plantlet. That plantlet will have root formation as well as it's a complete plantlet which can be achieved along with this direct organogenesis. Now you can also see in indirect organogenesis, what happens is when you inoculate the, let's say, leaf disc on a media, it will first give rise to all callus formation. This callus is basically unorganized mass of cells. This unorganized mass of cells, when we passage it or subculture it on new media which has cytokinins along with a little bit of oxygen, they will produce lots of adventitious shoots which arises from this callus, and these shoots will again proliferate further, elongate further to form a complete elongated shoots. Now these elongated shoots, when you separate it, excise it with the help of surgical blade, put that elongated shoots on an in vitro rooting media, you will have a in vitro rooting in the rooting media. And when this rooted and elongated shoots, when you transform from the rooting stage to a acclimatization stage, you will have a complete plantlet. So this indirect organogenesis is also possible in any type of explant, and it is also utilized for regeneration as well as genetic transformation of many plant species. So there are various factors which affect this regeneration and organogenesis. First is source of explant. Source of explant which is cultured is important in determining the potential of organogenesis. Most suitable plant part is basically a starting culture. Will depend upon, of course it depends upon the species, but it is found that leaf and leaf fragments of species of Begonia, Solanum, Nicotiana have shown the capacity to regenerate the shoot buds. So this kind of leaf tissues which are younger shoot has a capacity to produce the complete shoot formation. The next is physiological and ontogenic age of the explant. It is found that if that particular plant is actively grown, it is vigorous in nature, it doesn't have any diseases, it has been has a nutrition, or it has been fed with enough nitrogen source, those kind of explant will give rise to beta organogenesis. So apart from physiological age of the explant, the ontogenic age of the explant will also influence the organogenesis. What is meant by ontogenic age is a meristem age. And as you know that from the zygote, when there is a apical epicotyle formation and hypocotyle formation, that particular meristem is most juvenile tissue on the plant material, and therefore if you take age of the explant is as younger as it can be, the explant will give rise to more shoot formation. So that is called ontogenic age. And therefore the leaf explant collected from the vegetative phase prior to the flowering stage generally will give rise to more organogenesis response as compared to if the explant which is taken after flowering stage, because flowering stage is a maturity of the plant material or the mature phase of the plant, and therefore at maturity you will show less response, and ontogenically it is more more mature, and therefore if you take the plant material before flowering, and if it in fact is at the seedling stage, that will be more juvenile and it has a capacity to regenerate faster. Now the season in which explant is obtained, I have discussed this many times in all my session, that at least in India it was found that summer season in many cases because it shows active growth, at least in some tree species it is found that the explant will respond much better in summer season. Of course, in some plant species, if the rainy season or winter season where there is an active growth phase of that particular plant, especially in perennial plants, if the active growth phase starts from the rainy season or after the rainy season, then definitely that explant will also give rise to a high regeneration potential. But as I told you that the season influences surface sterilization, that is a different story, and season also influences morphogenetic potential, that is a different factor. So some seasons are favor say morphogenetic potential, whereas some seasons they favor the surface sterilization probability, the probability of not having contamination. Size of the explant also matters most. In organogenesis, it is found that the size, it is dependent upon the size of the explant. Large explant will have a higher regenerative potential and smaller explant will have smaller regenerative potential. Now let's discuss this with reference to thin cellular system. In thin cellular system, what we are trying to do is we are using in fact section of the plant material, so which is a smaller size explant. So this sometimes in a conceptually way it will contradict, but let me tell you that in some plant species thin cellular system has been extensively proliferated and it has shown a lot of result, but not in all the plant system thin cellular system has been so far worked out. So definitely size of the explant will influence in a larger context, or in a plant tissue culture wherever so far reports has been taken, it was always found that the larger size explant will have a more regenerative potential. Now culture environment or quality, in this quality and intensity of light, it was found that treatment of blue light followed by red light stimulate organogenesis, and this generally takes place in the illumination of 2000 to 3000 lux for 16 hours of light, 8 hours of dark. Whereas callus tissue of Nicotiana produces shoots or embryogenesis when the tissue is exposed to higher light intensity, that is you can look at 2000 lux and 10,000 to 15,000 lux. Typically one fluorescent tubes gives rise to 900 to 1100 lux light intensity, and therefore it depends upon from where you are basically measuring the intensity of light. If you take it from very close, it will be higher, but generally we take it from at least 2 feet or 1 feet away from the fluorescent light. So this light intensity is really high and in this light intensity there are some reports where you have got regenerative potential or embryogenetic potential from that particular callus tissue. Culture environment. In a cultural environment, temperature, it has been found that in some cases temperature, for example in anther culture we found that if you give a cooler treatment, pre-chill treatment to your explant, it has a high potential to form a haploid plantlets. Relative humidity also found to be influencing morphogenetic potential. The gaseous atmosphere within the cell, as I already told you that these cultures, when you plug the plug it with the cotton plug or with the plastic cap, there is a regeneration or development of ethylene gas, and this ethylene gas basically to certain extent induces necrosis or shoot tip necrosis or many other disorders. So lesser the ethylene gas presence, there are higher chances of morphogenic potential. As well as if you look at the physical form of the media, in case presence and absence of agar, I have already told you that there are certain micro propagation protocol of some species where the liquid media is found to be more beneficial, more stimulus than the semi-solid media. As well as pH of the culture media are also found to be showing much better morphogenic responses than the other types of pH. So these factors are found to be quite influential. Nutrient media as well as constituents. Plant growth regulators has also, as I already shown you, this hypothesis has been very well proven by Skoog and Miller, that the high cytokinin concentration will induce shoot formation and a low cytokinin concentration will induce root formation. And when you have intermediate concentration of cytokinin and auxin, you will have unorganized mass or unorganized callus tissue. This picture is very nice picture of showing those results. You can see that at a lower concentration of both carnitine and auxin, there is a very low response, whereas at the higher concentration of IA, you can see the root responses. A lot of adventitious roots are being produced from this particular explant. Intermediate concentration of carnitine as well as intermediate concentration of auxin. Carnitine is a cytokinin, indole acetic acid is auxin, and you can see the middle portion of this picture from where you can see the callus formation, a very proliferative callus formation. Whereas as you increase the carnitine concentration, you can see this is at the carnitine, and as you increase the auxin concentration at a higher carnitine and low auxin, you are seeing higher number of shoot formation, whereas as you increase, again there is a callus formation. So it is very clear that high cytokinin will give rise to maximum number of shoot formation, and high auxin concentration will have maximum number of roots on your explants. Now let us go to the next topic, which is somatic embryogenesis. And as I told you, this particular technology has such a tremendous potential, a significant potential to create mass multiplication of plantlet. It has such a huge capacity that some companies from USA and from Europe has in fact developed mass scale, a very large scale plantation industry by using somatic embryogenesis. Now what is it? It is a process of single cell, a group of cells initiating a developmental pathway which lead to the reproductive regeneration of non-zygotic embryos, and that's why it is called somatic. Non-zygotic embryos capable of germinating to form a complete plantlet. So there is no zygote formation in this particular process, no fertilization or unfertilized explants or vegetative explant will give rise to embryo-like structures, or in fact embryos, and these embryos further germinate to form a complete plantlet, that is called somatic embryogenesis. Now this somatic embryogenesis, this represents a very complete model of totipotency and involved action of complex signaling network. This somatic embryo development involves similar stages of zygotic embryos, which are globular shape, heart shape, torpedo shape, and cotyledonary stages in dicotyledonous species, whereas in monocotyledonous species you will see globular, scutellar, and coleoptile stages in monocotyledonous species. And once the somatic embryo reaches these cotyledonary or coleoptile stages, they will initiate a shoot meristem as well as the root meristem and the root formation, and that seedling growth will begin, or it will form a complete plantlet. There are two ways of somatic embryogenesis formation. One is called direct embryogenesis. Direct embryogenesis, where the embryos will form directly from cells or group of cells without production of intervening callus, and that is the direct embryogenesis. Indirect embryogenesis is where the callus is first produced from the explant and after that the callus will have embryogenetic potential, a regenerative potential, and the embryos will come on the surface of the explant or in between that callus wherever the meristematic clusters are present, or as well as from cell suspension cultures also. So cell suspension cultures will give rise to callus cluster will give rise to nodular callus will give rise to lots of embryos. These embryos will show lots of further proliferation to form maturation of embryos, and this maturation will give rise to cotyledonous stage will undergo germination to form complete plantlet. You can see from this slide beautiful, very nice stages of all the embryos, and these embryos again proliferate, germinate to form a lot of plantlet, small size seedlings, and these seedlings can be again further grow in a field or in greenhouses, and you can have tremendously large number of plantlet by using this technology. You can, if at all, if you have cell suspension cultures and those embryos develop on a liquid media, it is well and good. You can have a liquid culture, large scale liquid culture of somatic embryos, and these somatic embryos can be scale it at on a large scale with the help of bioreactors, and once you scale it at a bioreactors, you can have in fact millions of plantlets from this particular technology. Similarly, in indirect organogenesis also, as you can see that the explant will give rise to small embryos. These embryos will produce a cotyledonous stage, hypocotyle and torpedo stage, and all these embryos will further germinate to form a complete plantlet. So for all this organogenesis, again I have referred to book by Bhojwani and Razdan, Plant Tissue Culture Theory and Practices. If you have any questions, any doubts, any practical advice for all my lectures, or even for developing or establishing a plant tissue culture lab at your place, or if you need any advice with reference to your tissue culture project in your institution or in your college, you can always email to me or contact me. I would definitely like to help you. So with that, thank you.